Good afternoon. My name is Shawn Douglas. I'm from UCSF, and today I want to talk to you about a question that my lab is interested in, which is how can we precisely manipulate matter on the nanometer and even atomic length scale. And don't worry too much about this slide yet. I have an alternate title for my talk, which is this slide will make more sense in 10 minutes. And so I'll just keep this in mind. Before I get too much into how do we manipulate matter, I want to first talk about why. Why would we want to do this? And when we think about biological research, we have all these amazing tools that extend our human capabilities. So we can see all the way down to micro and nanoscale features of biological systems. But sometimes it would be really useful and helpful if we could just grab onto the things that we're studying and manipulate them and actually put them where we want them to go or pull on them. There's just a lot of things that we as humans do on the macro scale that we can't do on the nanoscale. And so that's what we're trying to do, is build the nanoscale hands to complement our eyes that we can, that we can achieve through these tools. So the next question would be, how do we actually build nanoscale hands? Because we can't do it sort of top down. And our lab is part of a growing community of researchers which is in the field of DNA nanotechnology. And so we use DNA to build our hands through self-assembly. And you might ask, why build with DNA? We usually think of this as a genetic information storage molecule. And the reason is that it happens to be one of the most simple and versatile, especially simple ways to realize programmable self-assembly. And the entire field was pioneered over the last 3.5 decades by Ned Seaman at NYU, and he recognized that DNA could be a nice simple building block to start using, kind of like Lego building blocks on the nanometer scale. And so then the next question is, how do you actually build with DNA, right? And let's just look at what the DNA structure looks like. It's a pretty famous molecule, so it's a double helix. And the first question we could ask is, how do we get from this linear polymer, this long chain, into two-dimensional and then later three-dimensional shapes? How do we build that? And the answer that Ned Seaman figured out and the community has adopted is that we can synthesize whatever DNA strands we want and actually encode cross-links between neighboring strands in the sequence information. So if you look at the first version of the slide, the two strands are separate. I can resynthesize some of these strands with different sequences such that they come together and cross-link. And this is really the fundamental, one of the fundamental operations that we can use to bring DNA together and to define structures. And this gets you like single-digit nanometer length scales. And so the next thing we want to do is actually figure out how to build larger structures. And to get to larger structures, we use a method called DNA origami. And DNA origami was invented a little over a decade ago by Paul Rodeman at Caltech. And he managed to achieve like 100 nanometer dimensions on these shapes. And I'm going to explain how this method works. It's actually really cool and fun to work with. And so we'll focus on this, the disk with 3 holes, as Paul called it in his paper. And what you'll notice about this structure is that it has hundreds of these cross-links. So it's much larger. And the way that it's designed is actually, it's built out of 2 types of strands. So there's one really long strand of DNA that can be 7,000 or 8,000 letters long, which we call the scaffold strand. And we know this sequence. We can actually make it in the lab using a variety of different methods. And then we also synthesize, or we order from a company, about 150 to 200 short strands of DNA that might be like 20 to 50 letters long, and we call those staples. And so the way that this assembles is in a one-pot reaction. We actually pool all of these strands together, and we mix them, and then we heat up the reaction. And so here's an animation that I made back when I was a grad student. And what we do is basically encode the information in those short strands such that when everything cools down, the short strands will find their complementary sequences on the long scaffold strand and actually cooperatively force it to adopt a specific shape. And the shape will depend on where we've put those crosslinks. So when we choose the sequences, we define the overall larger global shape that self-assembles. So we can typically make about 500 billion to a trillion shapes in the size of a raindrop, which is pretty cool. So self-assembly is quite powerful in terms of how many components you can make. And this raises the question, okay, what do you do with a trillion smiley faces or a trillion little stars, right? You can't really do much with that. They're kind of like artwork that represent the potential of the method. But if we return to biological questions and things that we're trying to study in the lab, we might actually be able to find some really useful things that we can do. And so I want to actually take a little detour and talk about another area of study, which is outside of my expertise, but I have collaborators at UCSF in this area of cryo-electron microscopy. And now this field is just about the same age as DNA nanotechnology, a little over three decades old. And thanks to development of direct electron detecting cameras in the last few years, it's really pushed the resolution of this technique to actually near atomic resolution. And so a few of my neighbors at UCSF in 2013, they solved this TRPV1 channel, and it's a really challenging target. Dozens of labs around the world were not able to solve it in the preceding years. They managed to get a high resolution structure, and you have this channel in your tongue. Everybody has this channel, and you recognize it as it's a heat and pain sensor. And it actually also can be wedged open by a peptide called caspacin, which is found in chili peppers. So we can now see on a molecular level, why do spicy foods actually taste hot? And which is pretty cool. So they also solved another related channel, the TRPA channel. And this one binds the wasabi pepper peptide. So we can start to look at, build molecular models and understand, how these different sensors work. But cryo-EM can also be applied to study viruses. For example, the Zika virus was actually, the high resolution structure was solved of the Zika virus last year. And so this method is actually quite powerful. And what we want to do is use DNA origami and this positional control to further extend and improve cryo-EM. Or we want to build hands that will complement these really powerful eyes. Okay, so before I can explain how origami will work, I'm going to do a quick 2-minute crash course so you can understand how does cryo-EM work, and then we'll be able to talk about how to overcome some technical limitations. So the first thing you do is maybe spend 3 years of biochemistry and prepare your sample, which is represented here as a duck. The Fourier duck. This will probably be the only thing you remember from this talk, by the way, so is the duck. But so that may take some time. Once you have your sample purified, what we do is we apply it to a special EM grid, which is about the size of your pinky nail. And what we have is basically a concentrated sample of our protein, and we want to a single layer of about 100 nanometers thick. And so we use an instrument called a Vitrobot. And it actually resembles this toy, the monkey with the symbols. And what it does is it blots away the excess liquid and then plunges the sample into liquid ethane to freeze it as vitreous ice. And so it really does look like that toy. And so this claps together and then freezes the sample. Once it's frozen, we can load this into the microscope. And once it's loaded into the microscope, we apply the electron beam and it basically projects 2D projections of the individual single particles onto this detector. Once we have this imaging data, we can pick particles out of that data, and then you'll see that we get all these different orientations. We can actually computationally line these up to make class averages, and then once you have different angles, you can use those to kind of back project to a 3D structure. And then you can refine and iterate this process in order to push the resolution of your structure. Okay, so that's the basic outline of the technique. Now, what are some limitations? First, and this technique is kind of a victim of its own success, the microscope time is very limited and expensive. A lot of people around the world are vying for very limited time on these microscopes right now. But that'll improve over time. Another is that small proteins can actually, they can be very poor contrast and difficult to pick and align. And there are also technical advances underway, for example, this Volta phase plate technology that may make it easier to see small proteins, but it would still help to be able to see where they are. Another thing that's challenging is damage to the sample at the air-water interface. So if the proteins are too high or too low and they're close to the air, then you don't get very good data. And another thing, and this really depends on the protein shape and characteristics of the protein on a case-by-case basis, but some proteins can actually just preferentially align all the same way. And so if you don't get a bunch of different angles, you can't really solve the 3D structure if you're only looking at the protein from one side. Okay, so if you remember back to the beginning, what can we do, or why would we want to manipulate matter at the nanoscale? I think this is like a perfect example of a bunch of technical challenges where if we just had hands and we could just grab onto this stuff, we could solve a lot of these problems. So for example, we might be able to, if we had hands, right, we could array everything really nicely and collect very efficient, densely packed samples, and so get more data out of our microscopes. We might be able to find the small proteins very easy if we put them where we know and we know where they are. We could move those proteins away from the air-water interface so everything is kind of at the same level. And we could also control the orientation of the samples if we had this capability. And so what we're working on, and for the last few minutes of the talk, I'll show you our progress so far, is how can we use DNA origami as our nano hands to overcome these technical challenges. And I should say that our colleagues in Europe are also working on related tools. So there's a nice paper by Thomas Martin et al. In PNAS from last year, and we're really inspired by this work as well. So we started by making a DNA origami picture frame that had a protein binding site in the middle. And we're validating this with a protein of known structure called a tail protein. It's a transcription factor. And the first thing we encountered was some really low yields of the protein binding. So less than 5% of our proteins, or our frames were showing up with a protein in them. And so one thing that we did was redesign the structure. And this is kind of nice thing about origami. We can redesign our shapes very easily. We designed this hinge structure, and here it is with and without the protein. And because we now have a flexible binding site, we think this is improving the yield. So now we have more than 90% of our hinges actually have proteins bound. It's hard to see, but I'm highlighting the ones that have the protein present. So there's numerous technical challenges, many more that I won't discuss, but going back to this goal, I just want to focus on this fourth one, overcoming these kind of preferential orientations. Now, because we're designing this structure, we have control over how the protein is arranged once it's bound. And so if you had your hand, you just want to twist the protein, right? So how do we do that with self-assembly? Well, we can look at the design, and if we zoom in on this DNA, you can imagine shifting the binding site by a couple of base pairs. So if we shift this upwards by 2 base pairs, the DNA is a double helix, right? So the protein's going to rotate by some number of degrees. And then if we shifted this down another couple of bases in the other direction, then we're going to get a different orientation. And so we can actually systematically designed several structures where we've shifted the binding site and we get different orientations predefined in the structure. And then we barcode them, and that's where the original slide comes in. Those tabs on the top and bottom of the hinge tell us which angle we're working at. So we can pool all of these together and measure them in one go. And so here's some preliminary data. We're still collecting more data, so these are each from only a couple of hundred particles per class. But you can see, we can make out the protein. Here's another view of that in a movie form. And we already have enough to mask out the origami part and actually focus on the protein and even just do a very preliminary 3D reconstruction. And now this work is still underway. And this is actually led by one really amazing postdoc in my lab. His name is Terrell Axel. He's going to be looking for a job soon. So if anybody interested to add this capability to your university, please let us know. And with that, I'll thank all of my group members, our funders, and thank you for your attention.